All right. 5.3 has been trying to tell the story of how DNA becomes a protein, because DNA holds the instructions on how to make proteins. And we started off right away saying that DNA is just an information molecule. It doesn't do the work of making those proteins. DNA is found in the nucleus. It's a double helix. It never leaves that nucleus. It has four nucleotides: adenine, thymine, cytosine, and guanine. Yesterday's lecture looked at transcription, how DNA becomes RNA. Because DNA never leaves the nucleus, we need an intermediate molecule that can read this message and kind of copy it, transcribe it, and then it can leave the nucleus and find a ribosome to make that protein. So transcription is going from DNA to RNA. And RNA is like half of a DNA molecule, and also the bases are a little bit different. You have adenine and uracil pairing together, and then cytosine and guanine pairing together. So today's lesson, we're going to focus in on RNA and how it becomes a protein through a process called translation. So this RNA is going to leave the nucleus, it's going to find a ribosome, it's going to use a lot of RNAs that we learned in yesterday's lecture to put it all together and make a protein. Okay, so yesterday we made that mRNA molecule by, oops, I thought I had a arrow here, unzipping the DNA, matching complementary to it, and then creating this mRNA molecule. Okay, so translation is the process of going from RNA to proteins. We're going to translate the code that RNA read from DNA. The code, remember, is 3 nucleotide bases for 1 amino acid. And these 3 nucleotide bases we actually call a codon. Okay? These 3 nucleotide bases are called a codon. So a codon codes for 1 amino acid. Translation takes place in the cytoplasm of the cell. Transcription, okay, the process that we learned yesterday, or the previous lecture, takes place in the nucleus. So now we're in the cytoplasm of the cell. It's going to involve all 3 types of RNA. Our ribosome, our tRNA, and something happened to my mRNA. Let me see if I can, I'll just copy this. Eek. Sorry. I'm gonna just make it smaller. Here. And our mRNA, okay? So our ribosome's made up of our RNA, okay? Each of these chambers is going to read a codon. This is my tRNA. It stands for transfer RNA. It transfers amino acids to the ribosome because what are proteins made up of? Proteins are made up of amino acids. Down here I have something called an anticodon. The anticodon is going to match complementary to the codon. And the mRNA is going to be read by the ribosome. So the mRNA is carrying those instructions for which amino acid is placed where. So mRNA was in the nucleus, okay, it was created through the process of transcription, and now it's going to leave the nucleus and enter the cytoplasm and be translated. So the steps in translation are a little bit more complicated. Number 1, our ribosome is going to attach to that mRNA molecule, and it attaches on a start code. All proteins start with AUG, okay? They just do. So we're just going to call it a start code. And then it's going to start to read each codon. As it's reading a codon, a transfer RNA or tRNA carrying that amino acid is going to match up and join to that mRNA molecule. And these tRNAs are just going to come with that amino acid, drop it off, and go find another one. The ribosome is going to keep moving along the mRNA while the tRNA matches up the amino acid and eventually links them together. After that amino acid is linked, the tRNA will fall off and go find another amino acid, and then the ribosome reaches a stop code, and everything just comes apart, and you're left with a protein. I know that may not make sense, okay? Words, sequences like this, you're like, I don't even know how this all happens, but you guys know me, and we're going to model this, and for some reason, I'm just going to do this. Do I want to do this? Yeah, I want to do this. All right. I just need a new slide. So I don't know why it carried off where it carried off. Sorry. Intermission. Play some music, right? Okay, so here we are. So here's my ribosome. This is my mRNA. These are my tRNAs carrying the amino acids. So these heads, or these circles, are the amino acids. The code reads, red's going to pair with gray, blue pairs with green. So here's my ribosome. And the first step in translation is that it's going to attach to a start site. So this is the start site right here. This red, gray, green. So now we know what the base pair in here, red pairs with gray, blue pairs with green. We are looking for a gray, red, blue. So I look, gray, red, blue. This guy right here. Comes in. The anti-codon of the tRNA matches complementary to the codon. Okay, ribosome slides over. Reads another code. Blue, blue, gray. So blue pairs with green, so we're looking for green, green, red. Green, green, red. Right here. Now, I'm going to do something like this. Something happens between these two amino acids. A bond is going to form. So the amino acid from the first tRNA is going to detach and form a bond. This tRNA. And the ribosome is going to move over. Now, as it moves over, you notice that this chamber is a lot smaller. Well, this tRNA is not going to fit because this chamber is a lot smaller. So it's kind of like an injection chamber. Okay, so the ribosome slides over, and this tRNA gets ejected to go find another amino acid. So now we have green, blue, red. So we are looking for blue, green, gray. Blue, green, gray is this individual right here. Or this tRNA, I should say. Now, these two amino acids form a bond with it. See, now notice my amino acid chain gets longer with each tRNA. Ribosome slides over. This tRNA gets ejected. And we're looking for gray, blue, red. This comes in. These amino acids form a bond with that. And let's just say this is the stop codon, okay? So the ribosome moves over, and everything kind of falls apart, okay? Just falls apart. Okay. And you'll notice right here are my amino acids. And when you get a whole bunch of amino acids linked together, you make a protein. We just made a protein. This is my protein, okay? And proteins are responsible for various things. The traits of us, eye color, hair color, skin color. Sometimes a cell is just making more materials within it, or maybe it's going to make a new organelle. I don't know, whatever. But we just made a protein. All right, so your assignment is going to look like something like this. It says transcription, translation, summary. And let me just go to a full screen here. Nope. There we go. So, step number 1, you're going to fill in the complementary DNA. This worksheet's going to put everything together. So I have one half of the DNA. Well, I'm going to match it complementary to the other half. And then we're going to use the top half. Okay, I know it says bottom. Sorry. We're going to use the top half. And from the top DNA, we're going to transcribe it into mRNA. All right? And then from mRNA, we're going to read the mRNA and figure out the anticodons. And then from the amino acid sequence and the mRNA sequence, we're going to figure out the amino acid sequence from the mRNA sequence. So let's just do number 1 together so we have no confusions on what we're doing for homework. Okay, I'm going to copy this. All right. And I'm going to put it into the notebook here so I can write and do it with you. Nice. And I'm going to lock it in place. Okay. Get a pen. Purple is fine with me. Here we go. Step number 1. Figure out the complementary DNA strand. So in DNA, A pairs with T, and C pairs with G. So A pairs with T, T pairs with A, C pairs with G, sorry, G pairs with C, T pairs with A pairs with T, G pairs with C, C pairs with G, T pairs with A pairs with T, A pairs with T, and we have G, G, and A. All right, we just figured out the other half of our DNA. Now we're going to make mRNA. Okay, in mRNA, A pairs with U, there's no T, but C and G remain the same. Okay, so A pairs with U, T pairs with A, G pairs with C, G pairs with C, T pairs with A pairs with U, G pairs with C, C pairs with G, T pairs with A pairs with U, A pairs with U, C pairs with G, C pairs with G, and then T pairs with A, T pairs with A. Okay, so we've just figured out our mRNA sequence, mRNA strand, or messenger RNA. Now we're going to do the anticodon, okay? So here A still pairs with U, and C pairs with G. Now before I move on, take a look at our mRNA strand, okay, that we just transcribed from this top strand of DNA. Notice that it's very similar to the one that you just wrote. In fact, it's almost the same, except our T's become U's. So that's why I wanted to do the top complementary strand so you could just, if you were really struggling, you know, just swap your T's for U's. And so this should be the same as what you do up here, okay? Okay, let's do the anticodon. So U pairs with A pairs with U, C pairs with G, C pairs with G, A pairs with U, U pairs with A, C pairs with G, G pairs with C, A pairs with U, U pairs with A, U pairs with A, G pairs with C, G pairs with C, A pairs with U, A pairs with U. All right, now this bottom part, we're going to figure out what amino acid it codes for. Okay, we're going to take it a step further. We're going to use something called a codon wheel. All right, so this is also included in your assignment. Let me find my codon wheel because it has disappeared from me. Where did it go? I just had it. Well, this is wonderful. Yep, here it is. Found it. All right, this is a codon wheel. How it works is this is the first letter, this is the second letter, and this is the third letter. And then it has the amino acids listed on the outside. So I'm going to take a look at my first three. I'm going to change my pen here. My first three, right there. UAC, okay? So first letter is U, second letter is A, and the third letter is C. So I'm going to write tyrosine, okay? Tyrosine. Wow, go me. All right, now we're going to take a look at the second set of three. CAU. CAU. histidine. Wow, that doesn't really look like histidine, but it's close. Now we're going to take a look at the third codon, CGA. So CGA, arginine. There. Okay, the fourth codon, UUG. UUG, leucine. And the final codon. Didn't think we would get there. GAA. GAA, glutamic acid. Glutamic acid. There. Did it. We just did number 1 together. We went from DNA to DNA, figuring out the other half. From DNA to RNA, just did transcription. Figured out the anticodon. So we went from mRNA to the amino acid, which is translation. So we put it all together. Isn't that incredible? All right. So with that being said, good luck. Let me know if you have problems. Don't hesitate to comment on Google Classroom. And yeah, that's all I got.